Okay, so another genetic question to be going through. This is about pea plants, and a couple of key things here. In my last video of question 5, I went through all the definitions we need to know. There's an interesting one here though, which is good to know, and that's pure breeding. And pure breeding would mean effectively homozygous. Okay, so one of Mendel's original experiments was to cure cross pure breeding. So the homozygous red flowering pea plants with pure breeding homozygous white flowering pea plants. The next year he grew the seed he collected. This first generation F1 of pea plants all had red flowers. Mendel then made each flower on these plants self-pollinate. He collected the seed from these flowers and then grew them. The second generation F2 gave the following results: 705, 224, and immediately that kind of screams to me about 3 to 1. The reason why it's not exactly 3 to 1 is that the exact 3 to 1 ratio is statistical and theoretical, whereas this is actually dealing with the probabilities on the ground. Okay, so which flower colour is due to the recessive allele? Well, the very fact that when the red was crossed with the white and the next year they all had red would suggest that red is dominant, and so red is dominant and white is recessive. So the recessive allele is white. Draw a genetic diagram to show the inheritance of flower colour in the first generation. Well, it says here that he took pure breeding red and pure breeding white, and they've asked for RR, small r and big R. So red would be RR and white would be RR. The gametes this could produce could only donate big R's. This could only donate small r's. If we Punnett square this, r small r comes to big R small r, big R small r, big R small r, big R small r. Red is dominant, so the phenotypes are red, 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 and red. Hence 4 to 0, red to white. And that's how they asked us to draw the genetic diagram to show the first generation of plants. Explain why Mendel made the first generations of plants self-pollinate. Okay, so now this one, I think this is a really good example of where to use the mark scheme, because I think this is quite a difficult one to explain. So if we go to our question, so the answers are as follows. To stop cross-pollination, okay, so that means that if they self-pollinate, there's not going to, pollen is in the air, it travels by air or possibly by insects, but there's possible contamination. But if you control the cross, you stop cross-pollination, okay, by crossing the offspring, okay. The second one on the mark scheme is to control the gene pool or prevent other genes getting in. That's the same thing. It's if you had cross-pollination, then you would have other gene pools contaminating it, okay. If you cross-pollinate, you are able to see which genes are present by the results, okay, which he subsequently did. And the last one is to test if the first generation or if they contained any genes from white or recessive genes, okay. So I have to admit, I think it's a difficult one to explain there, so I'm more than happy at GCSE level to go with the fact that the reason that they self-pollinate is to stop cross-pollination and to stop contamination from another gene pool. They're the two that I would put in, okay? So, if Mendel had taken any two of his white flowering peas and crossed them, what would have been the colour of the flowers of the next generation of plants? Well, if they're white, they're double recessive. So double recessive is RR times RR. And so, if you think about it, that's going to be pure small r's, isn't it? So they all would have been white. Okay. It's very difficult to get red flowering pea plants that breed true. Explain why you cannot guarantee to breed by self-pollination pea plants that only have red flowers. Well, the reason for that is that if you think about it, red is dominant. Okay, which means, going back to our definition of dominant, it will be expressed in the phenotype in either, do you remember that's the key word, the homozygous condition or the heterozygous condition. So you can't tell from the look of the plant alone, the phenotype, whether it is pure breeding, because there is no difference in the phenotype between big B and small b. And so you can't guarantee by self-pollination that you will have only red flowers, because if there is one of these involved and you cross-pollinate with another, then when they cross-pollinate, let's just do a quick Punnett square of that, which is actually similar to the one we've done earlier. You get big B, big B, big B, small b, big B, small b, and there is the double recessive, which is white. If on the face of it you've got all red flowers, but you don't know that some of them aren't that, then when you cross-pollinate, if you get two of the heterozygous, you're going to get the odd white flower. Okay? So the way the mark scheme deals with this is red is dominant, so masks any white alleles. Okay? It could be heterozygous, as I mentioned here. And then, therefore, a recessive white allele is present. There's a chance of a white flower being produced if there's a heterozygous cross, as I've done here. Chance of white flower could be 1 in 4. And they've gone into the ratio, and that is a 3-to-1 ratio, or 1 in 4. Okay? So that is a P question with Mendel, and we'll go on to number 7.